But for today, it's a great pleasure to have Emily Chapman speaking to us. And Emily's a postdoctoral position here at St. Mary's. You'll explain more, I'm sure, Emily, as we go along. Talking about adding something to reduce the hazards of something. Thank you for coming, Emily.
Thank you, Tony. And thank you all for showing up today. It's fantastic to see so many faces here. I'm really excited. And I'm very excited to talk about this project because the reason it's exciting is because, as far as we know, we're the first research team to actually investigate this particular question. That is the title of my talk today. So what we looked at was if a low-dose selenium additive could reduce the environmental risks of arsenic and mercury in mining waste. And this project was a collaboration between St. Mary's University, specifically Dr. Linda Campbell's, right there, Linda Campbell's research group, and our industry partner, Amick Foster Wheeler. Jody's representing them here today. And so the people involved on the research team, of course, was Linda Campbell, myself, and our very hardworking student assistant that's not here right now, but her name is Julian Robinson. And again, a person that's been instrumental for this project from Amick Foster Wheeler's side of things is Jody Berry. Other people from Amick Foster Wheeler that's been part of the research team includes David Parkinson and Chris Elliott. So that was the actual research team. And I should mention that this project was funded by an NSERC engaged grant, so it was a fairly short duration project. We did most of our field work and our lab work over a 6-month period. So the way that I've structured this presentation is I wanted to first give you a brief background on the gold mining sites in Nova Scotia and the environmental issues associated with the mining waste that's been generated at these sites. And then I'm going to move on to talk about selenium and why we think selenium might be able to alleviate some of those risks. And once you have that background, I'm going to move on and talk about our actual project and the different stages of the project and what we found. And I'm going to summarize all the conclusions and talk a little bit about where we see this future going, or where this research will go in the future. And hopefully, if you can get me to stop talking, by the end of that, we'll have some time for questions and comments. And I should mention up front as well that this is a fairly technical subject, and I've tried to keep it at a level that is of interest to everyone here because there's quite a wide audience. But if anyone is interested in more information and want more technical details, please let me know. There's lots of it. And I should also mention that we just recently submitted a paper this week to a journal, so we're hoping to hear back in a couple months' time, I guess, about the publication of it. So I'm happy to send along that as well once it's been published. So as promised, I'm going to start talking about the gold mining districts here in Nova Scotia. And there's 64 of them. And most of them were operational between the 1860s and 1940s. And the way that the gold was extracted back in those days was through a method called mercury amalgamation. And what that means is they took the gold-bearing rock, they basically crushed it in a stamp mill to a sand, almost silk-like material, and it's very fine. And then what they did was they spread this material over mercury-coated copper plates, and the free gold then combines with the mercury and can be scraped off these copper plates periodically. And then what you're left with is this pile of sandy, silty material that's also called tailings. And you'll hear me refer to that term a lot throughout this presentation, and what that basically is just mining waste. So what they did with the tailings, they usually just deposited them wherever it was, convenient. So in low-lying areas of the mine, like wetlands and even rivers and lakes sometimes. I wanted to show you this picture. This is actually the background to the poster of this presentation as well, so most of you have already seen it. But what this shows you here is a pretty typical tailing deposit. You can see sort of the sandy material here in the foreground. This one is I should mention here that this tailing deposit was deposited here well over 100 years ago. And there's still not a lot of vegetation on this one. Not all of them look like this. Some are more vegetated, some have trees growing in them and more plants. This one particularly is used by the public for things like dirt biking. The public love these areas. They're like big sandboxes to play in. And yeah, I just wanted to show you this picture because it's a pretty typical tailing deposit there. So it's been estimated that approximately 3 million tons of this tailing material was generated in Nova Scotia between the 1860s and 1940s. And the problem with the tailing material is, of course, it contains really high concentrations of mercury because of this mercury amalgamation method that I told you about earlier. And another problem is that it also contains really high concentrations of arsenic. And the reason for that is that the gold-bearing rock actually is really high in or rich in arsenic itself. So once it's crushed up into this little sand-like material, then it's concentrated. So those are the reasons why we have those contaminants in that material. There are other contaminants as well at some of these sites, depending on the extraction method of the gold that was used. So cyanide is an example. There's also lead and there's also copper, just to give a few examples. But the main contaminants of concern is, of course, mercury and also arsenic. And just to give you an idea for how high the levels are of these contaminants. In terms of mercury, we're looking at 140 times background levels, and for arsenic, about 340 times background levels. And of course, this varies across the different sites, but on average. So this, of course, is related to human and environmental health risks to have these high concentrations of contaminants in this material. And the risks that we're talking about, of course, it's risk to the terrestrial environment in close relation to the mine, but also to the aquatic ecosystems, close or even in estuaries further away from the mine. There's also risk of this material leaching into ground, or not the material itself, but the contaminants leaching into the groundwater, and also re-emissions into air of these contaminants. So why don't we just remove it? Why don't we just dig it out, treat it somewhere else, or ship it out and have it contained somewhere where it's more controlled? The problem with that is that the simple answer is just it would just be too expensive and very difficult. And another problem is, even if we were able to do that, disturbing this material even just a little bit could increase the risk of making these contaminants more mobile and making them leach out of this material, and we don't want that. And the third reason is that there's actually still gold in this tailing material, because back in the day the extraction process was not very efficient. So a lot of companies are looking at extracting this gold using more efficient new techniques. And also, of course, gold prices have risen recently. So that's another reason why this material just can't just be moved off site and treated somewhere else. So how can we manage these risks with this material at the site then? Well, in terms of human health risks, the first thing that comes to mind, maybe we could try to limit access to these sites. Or the problem with that is that, again, these sites are very big, and sometimes there are a lot of different owners. So part of it can be crown land, other parts are privately owned, and fencing it off is not really easy for that reason, but also because sometimes we don't even know where its tailings are. We have a fairly good idea, but these were deposited over 100, 150 years ago, so we're just not quite sure where exactly they are. One thing that has been done is that there's been warning signs put up at some of these sites that are used frequently by the public that I mentioned earlier. One of those sites is Montague Gold Mining Site, and another, I think it's Goldenville, yeah. But the public, as I said, really liked to use these sites. They're great big sandboxes to play in, and dirt biking is common in these areas as well. So there are other techniques that can be used, or technologies, I should say, at the sites to control the contaminants where they are. And I wanted to briefly talk about a few of those options. So for mercury, one of the technologies that are being researched a lot now is something called thermal treatment. So what that is they basically heat up the contaminated material to make the mercury vaporize, and then they catch the mercury that way. Other things that can be done is different leaching procedures. So you can add a reagent to the material to make the contaminants more mobile and leach out, but then you have to have fairly expensive engineered controls to catch the leachate at the other end. Otherwise, you can run into problems with this leaching into receiving water bodies. Another thing I want to touch on is phytoremediation. That is something where you use plants to suck up the contaminants from the soil, and then you can actually harvest the plants and get rid of the contaminants that way. And another advantage with having more vegetation at these sites is, of course, that that there's a lot of roots that start growing, and it binds this material into the soil and makes it less likely to move and get kicked up in the air. So there are lots of advantages with trying to revegetate these sites as well. The problem, of course, is that there's not a lot of plants that wants to grow in this because we're dealing with very toxic material. So the research that's being done is, what can we add to this tailing material to make it more likable for the plants? And so there's lots of that research going on. And then another thing I want to mention that you can do as well is that you can actually cover the tailings with a cover, so a clay liner or something similar. And what that does is that it stops leaching through this material with the rainwater, and also it stops contaminants from getting kicked back up into the air. So those are some of the things that you can do at the site. But the one thing that I wanted to talk about in more detail is something called in situ stabilization. And what that means is that you add a reagent to make the contaminants less mobile and less toxic. So you actually don't change the total concentration of the contaminants, you just change the form that they're in. What that would mean at these sites, you'd still have the gold intact for future recovery with a technology like that. The trick is to find a reagent that works on the contaminants present in the material. So for mercury, reagents that's been used for this purpose is sulfur-containing reagents and also organic compounds. And the reason sulfur is one of the compounds that's been used for this purpose is because it binds really hard with mercury, and it stops mercury from being as mobile as it was before. So as I said earlier, the trick though is finding a reagent that works on all the contaminants, because in this case we don't just have mercury, we also have mercury and arsenic and sometimes other contaminants as well. So just to give an example, sometimes a reagent can actually have a negative effect on another contaminant. So in terms of organic compounds that's been used to immobilize mercury, if you put that into an oxidized tailing, you can actually risk making the arsenic more mobile, and that's something you don't want to do. So again, the trick is finding something that works on all the contaminants present, taking into account the geochemistry and everything else going on at the site. So that moves us to talk about selenium, my favorite element. Selenium, and before I start talking about selenium and its relationship with mercury and arsenic, I'm going to first tell you a bit about selenium itself. So selenium is an element that was actually discovered by a Swede. I'm from Sweden. And it was discovered back in the early 1800s. And it's an essential element. We all need it. It's located very close to sulfur on the periodic table, so sulfur and selenium are similar in a lot of ways. And just like sulfur, as I said, we all need selenium. It's an essential element. The reason we need selenium is because it creates these selenoenzymes and selenoproteins that are very important to fight oxidative damage. So selenoenzymes are very important in organs like the brain that consumes a lot of oxygen. Selenium is also very important to regulate thyroid hormone levels and regulate the influx and outflux of calcium into cells. So we absolutely need selenium. There's no question about that. We would not survive without it. So the problem though is, like most elements, selenium becomes toxic at higher concentrations. And with selenium, that range between where it's essential and when it starts to become toxic is very narrow. So just to give you an example in terms of soil concentrations, because that's what we're here to talk about today, the selenium concentration required for plants to be able to absorb enough selenium for livestock is 0.45 milligrams per kilogram. But the concentration where we start seeing toxic effects of selenium is as low as 1 milligram, which is actually the CCME guideline value for agricultural soils. So we're dealing with a very narrow range here. And just to give you a little bit more background on selenium and soil concentrations, naturally occurring soil concentration, in Canada, selenium concentrations vary in soil between 0.3 and 4 milligrams per kilogram. So in some areas of Canada, like the prairies, for example, they have higher concentrations of selenium in the soil there than the CCME guideline value of 1. But Nova Scotia is not one of those areas. Nova Scotia is an area of Canada with relatively low concentrations of selenium in the soil. And other areas of the world with similar low concentrations of selenium include Scandinavia and New Zealand, just to give a few examples. Now we get to the interesting part here, because there's actually higher bioaccumulation of mercury in these same areas with low selenium concentrations naturally occurring in the soil. And a very famous example of that, I think everyone knows about bioaccumulation of mercury in freshwater fish here in Nova Scotia. That's a huge problem in Nova Scotia, or in Sweden and the rest of Scandinavia as well. And it's actually been related back to one of the factors that is impacting this mercury level in the fish is the naturally occurring selenium in the soil environment. And of course, there are many, many other factors that control the level of mercury in the fish, but selenium is one of those factors. And based on that observation, there was a study done in Sweden back in the early 90s where they added selenium to lakes to see what kind of effect it would have on the mercury concentration of the fish in the lakes. And what they found was pretty amazing. They actually found that the low concentration that they added into the lakes decreased mercury levels in the fish by 75%. And there was actually a more recent study done in China, sort of similar, where they looked at naturally occurring levels of selenium in the soil and compared it to levels of mercury in rice grains. And what they found was that with increasing selenium levels in the soil, they actually had decreasing levels of mercury in rice grains. So this effect has been attributed to the very high binding affinity between selenium and mercury. This binding affinity is 1,000,000 times stronger than the binding affinity between sulfur and mercury that I talked about earlier. And so what happens is that when mercury enters into an organism, because it loves selenium so much, it finds the selenium, binds to the selenium really hard, and renders these selenium enzymes that are so important in organs like the brain that consume a lot of oxygen. It renders those enzymes useless. So the toxic mode of action of mercury is actually it's creating a selenium deficiency. So what I'm saying is that if you are going to be exposed to mercury, make sure that you're also exposed to selenium at an appropriate level, because what the selenium can do is it can actually replace the selenium that's being taken away by the mercury. So this ability of selenium to detoxify mercury has been known for a long time. It was first confirmed in rats back in the 1960s. And since then, it's been confirmed in all sorts of mammals and other organisms as well, like fish and birds and even microorganisms. But what's very cool is that selenium doesn't just seem to have this kind of effect on mercury, but also a similar effect on arsenic. So selenium has a detoxifying effect on arsenic, and this has been proven in humans and in other animal species. And there's been a study that looked at, again, naturally occurring selenium levels in the soil and compared that to how much arsenic had been accumulated in plants. And they found that with increasing selenium levels, there was a decrease in arsenic levels in the plants. And there was also translocation of arsenic from the above ground portion of the plant to the roots, and that's also interesting. So this has been attributed to the formation of a selenium arsenic compound that's very easily excreted from organisms. So just to summarize here now, so what we found in the research before we even started this project, it was three things, and that's why we kind of got us started thinking about this project. So the first thing, it certainly looks like selenium is able to limit the bioavailability of mercury and arsenic already in the soil. Secondly, it certainly looks like selenium is able to promote excretion of these contaminants from inside of organisms. And finally, it also looks like selenium is able to promote translocation of these contaminants from the above ground portion of the plant to the roots. So these were the reasons we were thinking, wow, wouldn't it be interesting to see what kind of effect selenium would have in a contaminated material with mercury and arsenic. And that's why we chose these gold mining sites in Nova Scotia, because they, excuse me, they're the ideal in that the tailing material contains both of these contaminants at high concentrations. So this moves us in to talk about the actual project. So our three main questions that we dealt with during this it was a very intensive 6 months. We tried to fit in as much as we could with the time and budget that we had available. So what we looked at was three things. So the first thing was we looked at what kind of effect does sodium selenite addition have on the toxicity of mercury and arsenic in this tailing material to plants. The second thing we looked at was what kind of effect does sodium selenite addition have on the bioaccumulation of mercury and arsenic into worms in this material. And secondly, or third, what we wanted to look at as well was try to establish the most effective concentration of sodium selenite that would limit the mobility of mercury and arsenic leaching out of this material. So this is what we had to do during our 6 months, and let me tell you, it was hectic, but we did it. So now I'm going to move in and talk about our actual project and talk about each of the stages. So the first thing that we had to do was set up an earthworm culture. And this, let me tell you, was hard work. It took a long time, and it was a big, huge process. But we're now happy to say that we're one of the only labs in Atlantic Canada to actually have an earthworm culture, a research-grade earthworm culture, and we're very proud of that. And what it involved, so this is me mixing up the culture medium, and Julianne and I spent many, many hours in this dusty little lab mixing the soil for the earthworms that they were going to live in. And that's the soil where for the culture where they're happy. So it needed to be nice and, no contaminants or anything. So the first thing that we had to do was make an artificial soil mix. And that artificial soil mix was also our control soil for all of our toxicity tests for this study. The artificial soil was also the base for the culture medium. So that was the first thing that we did. And artificial soil is just a mix of silica sand, clay, and peat moss. And then you control the pH by adding calcium carbonate to get it just right, because you want to get it just right for the earthworms. So that was a very finicky process, and it took a long time, a lot of dust. So once that was done, this is the actual culture where the worms live. So so we ordered the worms from Environment Canada Toxicology Lab in Ontario, and when they arrived, they had to acclimatize to their new conditions. So this is in a growth chamber with controlled temperature, lighting, humidity, and the earthworms had to be in there for some time before we could actually do anything. And the earthworm species that we used for this study is E. Andre. They're a species that's used commonly for this purpose all over the world. And they're not very big. They're about this big. They're tiny little compost worms, and I guess this picture can't at least, they kind of look huge in this picture. Yeah, they're not very big worms. But of course, so the worms that were sent to us from Environment Canada are all research grade, meaning that they have been genetically analyzed and confirmed to be just the one species. Julianne and I got very attached to these little creatures by the end of the study. And it was really hard actually putting them into the toxic soil by the end of it. But yeah, we did what we had to do. So the first thing that we did was we actually did a reference toxicity testing once the earthworms had acclimatized. And what that does is that you have to establish the sensitivity of a new culture, and actually you have to keep that going if you're going to be doing toxicity tests. And you have to establish the range of sensitivity of your culture in comparison to what's normal and normal range for the species. So what we found was that our LC50, or the concentration where 50% of the worms died in a known toxicant, in this case boric acid, was very much on par with what was considered normal for the species in terms of Environment Canada standards. So that was good. That made us happy. And then what we also did was we looked at the survival of these worms in our control soil, so the soil that we mixed up, our artificial soil, and that was 100%. So that basically told us that we were ready to get going with our project. So the first thing that we had to do is we had to go out and get the tailing material. And the site that we chose for that was this Montague gold mining site, and it's located in Dartmouth, so fairly close to here. And I wanted to show you this picture, because this shows you an aerial over this area. So in this black circle here, you see that's the more recent tailing deposit, and that's actually the same picture I showed you earlier, that tailing deposit. So you can see how barren it is, and it's a large area. Now that's not the only tailing deposit at this site. There are other tailing deposits as well. So we did not collect our sample from this main tailing area. We collected our sample from this area here. We see this red little circle here. The reason we chose that area there over the more, easily accessible area here was because this area here had much higher concentrations of mercury. The reason for that is that that tailing deposit there is associated with a much older stamp mill that probably had slightly different extraction techniques. But we wanted the higher mercury concentration for the study, so that's why we selected the tailings over there. And at the same time as I was doing my sampling for this project, Julian Robinson also did sampling for her honors thesis, and what she did is basically she took sediment samples out here in this wetland that you can see here. She also sampled invertebrates, and then she analyzed these to see how much mercury had accumulated in these invertebrates. So that was done at the same time as this was going on. So this wetland, I should mention as well, drains into Mitchell Brook. And Mitchell Brook goes all the way across here into, I imagine this to be lower when I was, anyway, it goes all the way up through that black circle, so almost into the main tailing area there, and it empties into Lake Charles. So this is actually the sampling location where we collected my tailings from. And as you can see, it's a lot more vegetated than the first site that I showed you. So the tailings associated with this site probably deposited about 150 years ago, would you say, Linda? Yeah. So there was a layer of organic material on top of the tailings that we had to remove first. And that layer ranged in thickness between 5 to 15 centimeters. So we removed that layer first. And then what we did was we sampled the top 7 centimeters of the tailings, and we collected enough until we got about 15 liters of this material into these plastic-lined buckets. And then what we did was that we brought the buckets up to a big tarp that we had on the site. We dumped the tailings out and we started mixing. We mixed and mixed and mixed and mixed because what we wanted to do was we wanted as a homogeneous material to work with as possible for this project. It was a very, very important step of this. So we spent some time mixing here, and then we brought this back into the lab and mixed it again. And then what we did is we divided this pile into 6 sub-piles. And each of the sub-piles were treated with a different concentration of sodium selenite. So these are just our sodium selenite stock solutions. So we had 6 piles, and they were all treated with a different concentration. Of course, 1 pile we didn't add anything to, because we wanted just pure tailings. So once we had done that, we inserted the tailing material into these little flower pots with a little, this one, yeah. So basically just a glass fiber filter at the bottom to stop particles from dropping into the beaker that we had underneath. Because what we also wanted to do is we wanted to catch the leachate in this beaker. So that was the experimental setup, and this is what it looked like when the tailings were all in the pots. So just to walk you through what you see here. So we have 3 replicates of the artificial soil here. Then we have 3 replicates of just a pure tailing material, for lack of other. Yeah, so just a pure tailing material from the field, nothing added to it. And our lowest concentration of selenium was 0.5 milligrams of selenium per kilogram of tailing material. Then we had 1, 3, 8, and 15. So our range that we tested was between 0 and 15 milligrams per kilogram. So once we had done that, we let the tailings sit in that material for a week before we did anything, just to stabilize. And then we started our plant toxicity test. So what we did for that was, we didn't just use one plant species. We actually used a reclamation grass seed mix, so a mix of very hardy grass species. And the reason we did that was because to be honest, we were not sure we were going to get anything to grow in this material at all. So we wanted to increase the odds of at least something growing. So that's why we chose a seed mix and a hardy seed mix. So we added the same number of seeds to each of the pots of the seed mix. And we positioned them again in the growth chamber with controlled temperature, lighting, and humidity. And the pots were in there for 28 days, and we water the pots every single day. And at the end of that period, we ended up with this. So, and I think, just to tell you what you're seeing here, what we found in terms of shoot heights, the difference in shoot heights between these tailing replicates, it was just not significant. It wasn't significant. What we see here is the best growth in the artificial soils, our control soil, which was not very surprising. But we didn't just look at the shoot height. We also looked at the percent emergence of the grass. We also looked at the root lengths, and we looked at the total biomass. And something else we looked at was chlorosis, which is basically yellowing of the grass shoots. So it's insufficient production of chlorophyll. So we looked at all those parameters, and to do that we actually had to harvest the grass. So we very carefully removed the grass from the pots, and we inserted them into these glass vials. And I apologize for these not being in any particular order now, but I'm going to try talk you through this to tell you what you're seeing here, because I think this picture shows a slightly different story. So again, I thought this was going to be much lower, but I'm going to... So at the very top corner up here, where it says 0 milligrams of selenium per kilogram of tailing material, that grass in those 3 vials there grew in that pure tailing material, where we had not added any selenium. Our lowest selenium concentration was here, 0.5, and then we have the next one in the middle there, 1, 3 way over here, 8, 15, and then we have the artificial soil here. And if nothing else can be gained from this picture, I think what you can see here is that we definitely had the least biomass in our pure tailing material up here in the corner. And if you don't believe me, I will show you this graph. This nice colorful graph. So this shows percent emergence of grass shoots in the pots in each treatment each week. So you have the percent emergence on the y-axis, and you have the different treatments on the x-axis. So what you see here again, the artificial soil, we had new seed or new shoots popping up every week, even the last week. That was not the case, again, with our pure tailing material here, the 0 there. After the 3rd week, we actually noted that the grass started dying in those pots. Sort of similar results for the lowest selenium concentration, but then something happened. And I want to draw your attention to this particular concentration right there. 3 milligrams of selenium per kilogram of tailing material. At that concentration, the percent emergence of grass shoots was actually the same as we had in our artificial soil. And that's a very, very exciting finding of the study. But I also want to show you, because we didn't just look at the percent emergence, we also looked at root length and biomass. And what we found was that root lengths, biomass, and percent emergence all increased with increasing concentrations of selenium, and they were all significant relationships. So that was a very exciting. We're very excited about this. So basically, we have better growth in the pots with more selenium in them. And I also want to show you this graph, because as I mentioned, we also looked at chlorosis. And what we found, and what this graph is showing, really quick, is that we saw greener, healthier grass in the pots with more selenium in them. Just to summarize this graph, what that means. So that was our plant toxicity test. So we're going to move on now and talk about our leachates, because as you can remember, we the leachate that was collecting in here we wanted to look at as well. So at the end of the 28 days, when the plants were done growing, we basically took the leachate that had been dripping into, because these, as I said, these pots were water every day with RO water, so we had water dripping into the beaker underneath. So we took the leachate and we had it, well, first of all, what we did was we filtered it to filter out the particles in there, because we still had some particles despite this filter that we had at the bottom of the cup. So we filtered out those particles, and then we had that leachate analyzed for dissolved arsenic, mercury, and selenium. And this graph shows you the amount of arsenic and mercury. So we have arsenic on this axis here, mercury on this axis here. I'm going to look at this graph first, and then selenium along here. So what we found was actually a decrease in both mercury and arsenic concentrations, with increasing selenium concentrations in the tailings, up to a concentration of 1 milligram of selenium per kilogram of tailing material. After that concentration, as you can see here again, we started seeing an increase in mercury and arsenic levels. They started to increase again, but they never really reached the level in the untreated tailing material. But just to show you where this decrease was significant, I've enlarged that rectangle into this graph here. And so basically this is the non-treated tailing material, and this is our most effective selenium treatment level, so the 1 milligram per kilogram. So the difference in mercury concentrations between the non-treated tailing material and the 1 milligram level was 94% in this leachate. The difference for arsenic was 74% with this particular selenium treatment level. So what we also did, what we wanted to look at, was how much selenium was leaching out of each of these treatments, because of course we don't want selenium leaching into the receiving water bodies, because at a higher concentration selenium can become toxic, so that's something else that we looked at. So what we found, not surprisingly, with increasing selenium additions to the tailings, we also had increasing selenium in this leachate. Now the increase was not very big here in the lower concentration range. There was actually even a decrease between the 0.5 level and the 1 level. And this is an important finding, and I'm going to get back to this when I talk about the conclusions a little bit later, so keep this in mind. But I want to tell you first about our worms. So the main purpose of the worm test was to look at how much mercury and arsenic had accumulated in the worms. We were not so concerned with the toxicity, because we knew this was going to be toxic. This is toxic material we're dealing with here. So we were more interested in looking at the range of accumulation of these contaminants with the different selenium treatment levels. So the way that worked was we took, after the grass was finished in here, we took the same soil, took it out of there and plunked it into a glass jar, and then we added 10 worms to each of the jars, and then we let the worms sit in there for 8 days. The reason we chose 8 days, it's not like an arbitrary number we just came up with, there's a purpose behind that. Like I said earlier, we really wanted to keep as many worms alive as possible in these treatments, because we wanted to analyze them at the end of the test. That was our main purpose. We wanted to look at the concentrations of mercury and arsenic. So we did a little test before we did our definite test, where we tried to see how long the worms would survive in the pure tailing material, and it looked like after 8 days they started to die off. So we wanted to keep the length of this test to 8 days, because what happens when worms die in this material, they just turn into mush. You can't really scrape it off, and you can't really analyze the dead worms. And also, a dead worm does not accumulate the same as a living worm. So our main purpose was trying to keep them alive in all of these for as long as possible. So after the 8 days, we found something very interesting that we didn't expect, sort of a side find, but very interesting. In the tailing material where we hadn't added anything, so no selenium, in that tailing material, the worms showed some pathological symptoms. Some of them had decomposing tails, like you see in this first picture here. Others were, I realize now these are kind of blurry, so it's hard to see, but they were sort of swollen and blistery, the worms. And we did not see any of these type of pathological symptoms in the selenium treated material, regardless of concentration. which I think is an interesting finding. It wasn't really part of the study, but I just wanted to mention that before I go and talk about the arsenic levels in the worms, because that was one of the purposes of this study. So what we found was a significant decrease in arsenic concentrations with increasing selenium additions up to a concentration of 3 milligrams of selenium per kilogram. After that, the concentration in worms started to increase again, as you can see here on this graph. And this graph is just this enlarged, this little rectangle enlarged here, so you can see that relationship. And before I start talking about the mercury in the worms, I have to show you this table. Because I'm sure a lot of you people now are wondering what kind of concentrations of mercury and arsenic did they actually have in the tailing replicates that they were looking at. So what we have, and I'm going to try to explain this graph, or this table. So over here, the first three, the sample IDs, starts with AS. They are our artificial soil samples, so we can sort of ignore them. They're nice and clean. Then the rest of the samples here, the T samples, are all our tailing replicates. And then you can also see the level of treatment in each of these replicates, the level of selenium treatment in each of those replicates. And I want to draw your memory back to that picture of Julianne and I mixing on that blue tarp. So our main purpose for that, of course, was to try to make this homogeneous. And we did a fairly good job in terms of arsenic. There's a little bit of a difference there between different samples. But we failed for mercury. And this was something we didn't realize until we had this analyzed, of course. So this was a little bit after the fact. But this, as you can see, the lowest concentration of mercury that we had in any of the tailing samples were actually in one of the non-treated tailings here, the T1s. That was 28. That was our lowest, while our highest was as high as 175 milligrams per kilogram of mercury. So there was a wide range of mercury. So to start with, we didn't actually see any kind of relationship with, our added selenium in terms of mercury changes in the worms. But once we found out about this, we were able to do a multiple regression analysis, taking this confounding. We suspected this was a confounding factor. And with that done, we actually were able to find a significant decrease of mercury inside the worms with increasing selenium in the tailing material. So now I'm ready for time. So now I'm just going to quickly summarize the conclusions of the study. So what we found, without a doubt, our sodium selenide addition decreased the toxicity of the tailing material to the plants that were growing in the study. And that's a really important finding because selenium additions could be a very cost-effective way of, helping to revegetate these sites. The second thing I want to mention is that we did find a decrease in both mercury and arsenic levels in the worms with increasing selenium additions. For arsenic, that relationship was only significant up to 3 milligrams per kilogram. And then again, our third, if you remember those three questions that we were looking at, the third one that we wanted to answer was the most effective concentration of selenium to try to limit the mobility of mercury and arsenic leaching into the cup here. And that concentration we established in the study was 1 milligram of selenium per kilogram of tailing material. At that concentration, the amount of mercury in the cups decreased by 94%, and for arsenic that decrease was 71%. And those two were significantly different from our control. Now, as promised, I'm going to go back and talk about the selenium leaching out together with this mercury and arsenic that I mentioned here earlier. So at that particular concentration, the 1 milligram of selenium per kilogram of tailing material, we had a selenium concentration in the leachate of 10. Comparing that to the selenium concentration we had in the leachate from the non-treated tailing material, that was 3. So that we had about an increase of about 7 micrograms per liter in that particular treatment. So now the question is, what kind of effect does that little bit of an increase in selenium concentrations have on receiving water bodies? I'm not saying that the 10 micrograms per liter is the resulting concentration that's going to end up in the receiving water body. Probably not, because that's going to be dependent on the catchments. I mean, we're talking about this little water collecting here in the cups, right? So that'll be dependent on the catchment area and the geochemistry and everything else. But there will probably be an increase in selenium leaching into the receiving water bodies. And the question is, will that have an adverse effect? Could it potentially have a beneficial effect? And the reason I mention that is because I want to go back now and talk very briefly about what Julianne found with her. That's why I mentioned her sampling in that wetland. She found that the concentration of mercury there in the sediments was actually as high as 350 milligrams per kilogram. And she also found that that mercury is bioavailable. It's accumulating in the invertebrates there. There were significantly higher concentrations of mercury in the invertebrates in that wetland compared to invertebrates in a different wetland. She's going to present this research. I think, is it in April? Yeah.
Yes, and there is a reference site upstream, which was much lower, but the sensitivity of the area itself is the same.
Yeah.
Yes, and that mercury is from the wetlands.
Yeah. So this brings us into talking about future research directions for this project, and that's definitely one I want to look into as well to see, maybe this little bit of an increase in selenium will have a beneficial effect on receiving water bodies. Secondly, what I wanted to do, which I think is very important, right now with this pilot project, we just did a very wide range here. We tested 0 to 15 milligrams of selenium per kilogram. I think what we managed to do with this pilot project is we narrowed it down to where the selenium is most effective. And I think it would be very interesting to delineate the effects in this lower concentration range of selenium. Maybe using different tailing material to see, with different geochemistry, if selenium is as effective. And so that would be one of the things. The other thing that would be absolutely necessary is looking into cheaper sources of selenium, so commercially available sources of selenium that can actually be spread across big mining sites, because in this study we used a lab-grade expensive chemical, and that is just not going to, that wouldn't be a cost-effective way to treat this. But what would be a very cost-effective way, potentially, of dealing with these risks is if we can find commercial sources of selenium that will do this. And I have a few of these sources in mind, and I want to mention one particularly. In Finland, because the Finnish people, they actually discovered a few years back that the Finnish people are selenium deficient. So they started adding selenium fertilizers to their fields to up the selenium level in their food. And that would be one thing, that there's selenium fertilizers out there that's significantly cheaper, cheaper than what we used for this study. So that would be worth looking at. And the second thing that I think would be very important, and I really regret we didn't have time to do with this project, was look at the speciation. What's happening? What's the mechanism behind this? We need to figure out how stable these contaminants are long term with the selenium additions. That's a hugely important part of this, and I really regret we didn't have budget or time to do it for this very short project, but we did what we could. And then of course field trials will need to be done as well to see how effective this is in the field, and perhaps doing some kind of mesocosm where you mimic field conditions to get an idea for if more than one application is necessary over time and so on. So these are just some of the future research directions that we see this potentially going in, and I'm sure there are many, many more. But to do this, of course, we need more funding. So Linda and I are currently looking at different options for different research grants, and a lot of these research grants require an industry partner, and it's a pretty good deal for the industry partner. Sometimes there's not even a cash contribution necessary, just in-kind contributions like time and equipment. And when there's a cash contribution, sometimes it's also eligible for tax benefits. So it's a pretty good deal for industry, and I definitely think that we need industry on board on this, because it's one of those things, one of these projects that will benefit from that. And if anyone's interested in collaborating with us, please contact me. I have provided my email address here, 2 of them. And I think this sort of concludes, and this leaves us some time for questions and comments if there are any. Yes?
from working with Mike Parsons in that area and getting him to do sampling for me, I think I know why your mercury concentrations were not. So mercury binds to the organic matter. So you took that off and then you were doing the next layer down, which I believe is the layer that actually leaches from the top.
Ah, yeah.
So it depended on how much little amount of organic matter you were getting in your mix.
Yeah.
So I don't think you can, if you really want to look at mercury and arsenic, you can't use the same soil, right? Because for mercury, you're going to have to look at the more organic end, because mercury stays on the surface in the organic layer, while arsenic gets leached out of it and moves down. So I think you have to, that's one big thing you have to
try to separate,
Yeah, you can't do them together.
And that's the other thing, speciation and what form they're in, that is really lacking in the study, and that's something that, again, this is just a pilot project, and we did what we could with the time and the.
Oh yeah, no, but I'm just saying, you need to do them, unfortunately, separately.
Yeah.
And see how, because your mercury results, is pretty low organic matter in your test's tailings.
Yeah.
So would it act differently if there's a higher organic? So
And I should mention that we did actually analyze the organic content of this material, and it was similar in all the treatments that we had.
Okay.
It was all the same, more or less, like.
Yeah, but I think that sort of explains. As soon as I saw it, I went, well, it's from where you got it. Like, I would do sort of, if you're going to pick a place, sort of look at sort of some kind of horizon sampling first to get a better idea.
No, and that would be interesting to look at the different, for sure, and necessary as well in this case, to move forward to do that. Yeah, I agree. Yeah.
Your material in your test is quite mixed and loose, so have you considered what the impacts might be on material in the field that would be a little bit more consolidated?
You mean like
in some of the tailings facilities that I've been to, or tailings areas that I've been to, it could be almost like concrete at the surface where
like
there's
hard
no water.
pan. We haven't considered that because, again, we didn't do that with this study, but it would be interesting to see, of course, what kind of effect it would have in that type of harder material, because there's a lot of hard pan at Montague, at that main tailing deposit there. We didn't sample in that. But yes, that would be another thing to look at for sure.
You said you used a C mixture for your treatment?
Yeah.
I was curious if you noticed any differences between species, between the different treatments, or if it's basically the same ones coming up each time?
Well, we didn't really look at the species, which species came up, but they all look the same to me. I don't know. No, we didn't look at it. That would be really interesting if you could distinguish between the species that popped up. That's brilliant. Yeah, I wish I'd done that.
So I was curious about your soil pH in your study area, and I was wondering if soil pH had an effect on selenium's affinity to mercury.
So the pH that we had in this tailing material was around 4, and it didn't differ between the different treatments, even with the added selenium, and didn't change significantly with the selenium efficiency either. So it was pretty much stable for, and it's interesting because most of these tailing deposits in Nova Scotia from these old gold mining sites it's not acid, but the tailing material is not acidic. It's around 5 or 6, but in this case it was slightly more acidic, so 4 was the range there.
One of the things that we found on some tailing sites that have that low pH problem, the pH alone can really influence the growth of the vegetation.
Definitely.
And by adding nutrients, of course, the soil's been stripped of all its organic matter and nutrients because of the low pH in some cases. And by adding some nutrients and finding the right mix of nutrients, you can enhance the vegetation growth. But the other thing I think you should be mindful of is that selenium leachate issue is really a critical issue when looking at potential implications for the receiving environment, for definitely in a wetland situation, because the speciation of selenium kind of supercharges in wetland systems and will accumulate quite rapidly in biota in those systems and can have effects both for skeletal effects in fish and fishing wildlife. So that's a good thing to
Exactly.
really
That's
understand.
why, that's one of the future things that we
Yeah.
need to look at as well.
Your work seems to show that the optimal concentration of selenium was between 1 and 3 milligrams per kilogram.
Yeah.
But on one of your early slides, you put up the CCB guideline for selenium in soil.
Yes, thank you for,
Yeah, so I'm just wondering about, are you concerned about
yes.
exceeding the guideline?
Yes, that's another thing. That's another thing we need to consider, of course, because we don't want to add, something that's. But at the same time, when we were thinking about what range should we test here, we didn't. This was just again a lab thing, so we didn't do anything in the field. But what we didn't want to do is, because we knew we had to look at a very low range. That's where we want, the effect to be seen. But what we also didn't want to do is not add enough, because we were dealing with such high concentrations of mercury and arsenic, and we were thinking that maybe we needed a higher concentration of selenium to bind all that, because, and that's the reason we tested a higher range than the, and thank you for asking that question, I should have mentioned that actually, it's a very good question. So that's the reason why we tested a wider range, does that make sense?
Yeah.
Can I just, sorry, being a regulator here,
Yeah.
explain about the guideline? So maybe, so you probably took the generic guideline, which is the lowest of both the environmental and human health.
Yes, I did.
So I don't know which one it was. It was human health. So the ecological one might be quite a bit higher.
I'm pretty sure that it was
Ecological.
ecological,
It was
because the
ecological?
plants, ecological.
Okay.
Yeah.
Okay. So just wanted to be
Yeah,
sure.
yeah.
And okay, so selenium works by sort of replacing sulfur in the body. Is that what you were saying?
No, selenium is part of special enzymes that work to protect against oxidative damage. So it's kind of similar in sulfur in a lot of ways as well, because we all need it. And it also, so what selenium also does is it regulates thyroid hormone levels and the level of calcium inside cells. So that's the function of selenium in the organism.
Okay, so it doesn't compete with
No.
sulfur. No.
The question around the public's interest in this. So you mentioned early on that it's a popular site with off-road vehicles or bikes, this sort of recreational use. And so I was wondering, are there any groups that are concerned over this toxicity, either amongst the environmentalist community or neighboring landowners?
Oh, yeah, I think there's a lot of, there's actually, I know there's a study being done, is it in Queens, about risk perceptions of these sites? And I don't know when, it's a master's thesis that's being done. Can you talk more about that?
Yes, there was a strong action to do communication to the public about the risks of these sites, and that's been happening. There's been advertisements in newspapers, articles, workshops, town halls, you name it, it's happened. The people who enjoy ATV, regardless of that, will still go. And if you can see the sign here in the picture, the government put up a lot of signs saying do not access these areas. People took those signs down.
They also shot
and they all,
Yeah.
yes, and this one is very high up in the tree, which is why it still remains very bright. So people are aware of these issues, but they value the use of that land more than the risks that are apparent, or they don't clearly understand the risks of the area. And it's a work in progress.
So there's not great deal of concern over these sites in relation to their own health?
There is concern. It depends on which group you're speaking with.
Yeah.
We should wrap it up at this point. And yes, I was going to ask you about that. So two things. First of all, I hope you'll join with me in thanking Emily for a fascinating talk. And second, Emily is preparing publicity for next week, where Linda Campbell is giving a talk, as you can see, on heavy fish. Same time, same place here next week. So you're most welcome to that.